My name's David Farrell. I'm the University Archivist here at Cal in the Bancroft Library, and it's my personal pleasure to welcome you to this session on biotechnology. We're calling it Biotechnology and the Biological Revolution, which we know figured large at Berkeley in the Bay Area. And interestingly enough, it figures fairly large in the Bancroft Library collections, which I just want to say something about for a minute. The focus of our collecting at this point is the program in Bioscience and Biotechnology Studies, which Sally Hughes, one of our speakers today, is the head of. And I worked with Sally a couple of years ago in its earlier incarnation, the program in the history of the biological sciences and biotechnology. And the focus of that was oral histories of documenting the Bay Area, documenting bioscience and biotech in the Bay Area. And I knew about Sally's work and how expansive and wonderful it was. And along with that had come some archival collections. The collections of Gunter Stent, who'd been a professor at the university and had corresponded widely with many of the leading figures in the early days of molecular biology. But I wasn't really sure about how much else we had. But in the years following, and since I've looked at the collections, I'm happy to say we have a nice collection, a really respectable collection in this area already at Berkeley. I think the kind of pillars of it, the landmarks of it, were the collections of the Nobel laureates Melvin Calvin and Wendell Stanley in virology, Melvin Calvin in chemistry. But those were really landmark collections as well that have helped, that gave us a foundation for collecting this area. We also have a number of departmental records, and college records. We have chemistry, College of Chemistry, we have agriculture, we have biological materials from different agriculture as well as different biological collections, and some very interesting collections documenting the reorganization of the biological sciences at Cal, which were really kind of landmark events themselves with implications beyond Berkeley. The earlier one when Wendell Stanley arrived and sort of threw his weight around as the Nobel laureate and the way he wanted things and the negotiations he'd made with President Sproul, including the construction of a whole new building for virology. And then later in the mid-1980s, when the biological sciences, and many of you were aware of that or participated or knew about the reorganization, and that was a major, major event. So we have materials related to that as well. And then in the fullness of time and working with Sally and others and being in touch with people, we've in the meantime collected such papers as Bruce Ames and Horace Barker and Dan Koshlan. So I'm happy to say we have a nice collection, and it's growing. And I think the major, on the other side, the industry side, the major collection is the records of Genentech, which we may hear a little bit more about today. So as far as our afternoon, we have three speakers. Sally Hughes, who is educated at the University of London and at Berkeley and the University of California at San Francisco. And she is the historian of science and director of the program in Bioscience and Biotechnology Studies here at Cal, and has been very active for many years, 15, 20 years, in documenting oral history, and is focused on particularly the scientists at San Francisco, at Stanford, and at Berkeley. And then also did a landmark series on AIDS, which is really not that connected with Berkeley at all, but she was doing when she was at San Francisco. And presently she has a book in progress on the history of Genentech. Our second speaker is Daniel Koshlan. He was educated at Berkeley and University of Chicago and worked at Brookhaven National Laboratory. He returned to Berkeley in 1965, where he has been professor of biochemistry and molecular biology. I thought this was a fascinating thing about his resume. He demonstrated that bacteria have short-term memory. And I'm sure that has great implications for us, but that's wonderful. And his recent work has emphasized the chemical mechanism of memory, the structure-function relationship of receptors, and the catalytic power of enzymes. And our third and final speaker is also a name that's well known to Berkeley audiences, although he's not a Berkeleyan, Professor Daniel Kevlis, who's the Stanley Woodward Professor of History and Adjunct Professor of Law at Yale University. His research interests cover the history of modern science, including the history of intellectual property in living organisms. His books include The Code of Codes, Scientific and Social Issues in the Human Genome Project, and In the Name of Eugenics, Genetics and the Uses of Human Heredity. So with that, we'll move on to our speakers, and we will hopefully conclude before our hour is up, and we'll have time for hopefully a few brief Q&A at that point, but we'll start now with Sally Hughes.
Thank you, David. It's actually been close to 30 years. My entire career has been at the Bancroft Library, so I feel very privileged to take part in its centennial. I doubt I'll have another chance. I'm actually going to deviate a little bit from the program. There. by spreading this out a little bit beyond bioscience and biotechnology. Professor Starr gave a wonderful talk this morning, and he mentioned Herbert Howe Bancroft's effort in collecting oral histories of California and the West. In 1954, the university again took up that torch by founding the Regional Oral History Office, which for most of its 50-plus history has been housed in the Bancroft Library. The Oral History Office now has something like 2,000 in-depth oral histories to its credit, documenting most recent areas of endeavor significant to the American West. Subjects range from politics and jurisprudence to wine and the culinary arts, to the history of the university and our own faculty. In the past, these histories have been produced as transcribed, edited, and indexed volumes bound in the traditional blue and gold colors of the university. Currently, the oral histories, with permission from the narrators, are also being posted online in fully searchable format, and hence can be accessed by virtually anybody within reach of a computer. If you Google Regional Oral History Office, you will find a pull-down list called Featured Projects, which will lead you to the online oral histories in each category. Today, I will focus on just one of the programs of the Oral History Office, the program which since the early days has documented the history of recent science, technology, medicine, public health, and health policy in something like 250 oral histories, with almost 150 on bioscience and biotechnology. But before we provide a few audio and video examples, I would describe very briefly why one would want to do oral history of science. You might ask, don't scientists publish papers, write up experiments, and give presentations? Isn't science already more than adequately documented? Aren't we in fact overwhelmed by scientific literature? Well, yes and no. Yes, we are overwhelmed. No, the formal literature does not adequately document the full breadth and context of science. Scientific publications aim to present the facts of research findings, but they tell you little, if anything, about the context in which the research was done, and the motivations, character, and worldview of the people involved in it. Oral history is an excellent method for getting at just such elusive information, information that helps to explain why events evolved as they did. A very short list of the sorts of information that oral history is good at recording, and which almost always are left out of scientific publications, might include the following: how the scientist's family, background, and education influenced his or her career choice and research interests, the conceptual thinking informing the research, why the research was structured in the manner it was, the nature and extent of individual contributions to key experiments, the influence of the national political environment on funding priorities, and on and on and on. Well, you might say, that is all very well and good, but isn't oral history a highly subjective method for doing history? Yes, emphatically yes. It is subjective. A major objective is to capture the personal and idiosyncratic viewpoints of significant actors in historical events, usually with results that are difficult or impossible to find in other sources. The major caution for the user of oral history is not to take anything at face value, but rather to use various types of sources, archival as well as oral, and each with its strengths and weaknesses in conveying the elusive historical truth. And now we'll turn to 3 audio clips with the help of, very necessary help of Alexander, and we'll end with one video clip. The first is a clip from a very long oral history with Charles Towns. He, as many of you in the audience will know, received the Nobel Prize in Physics in 1964 for inventing the maser, the forerunner of the laser, which he conceived and later built at Columbia University. He was appointed university professor at Berkeley in 1967, but of course we like to claim all of him for ourselves. Here he is talking to my colleague and the interviewer, Suzanne Reese, about scientific intuition. The extract provides a glimpse at the workings of a scientific mind. It's not happening? No. The technical problems that I feared. Well, I will read it. Reese. And what about intuition? Towns. Well, intuition is important. There's something still a little different. One aspect of a good understanding of physics is just to feel completely at home with all of the processes and the elements of the world around you. To feel they are part of you. They are good friends. You know exactly how they behave. I think of atoms and molecules and electrons in them, and I have a picture in my mind. I can just see the electrons and how they are moving and what they would do. If somebody asked me, now what would happen if a molecule does this and that? Well, I just think about the molecule, just as you would think about a friend, and what he would do in a case like that. You have then such an intimate knowledge that you don't have to write down equations and figure things out. I would call some of that intuition. You just say, well, you know, this is the way it has to happen, because that is what the molecule is like. You know the personality of a molecule. That's what it's going to do, that's what it has to do. Generally, if it is an important question, you might want to write down equations and figure it out mathematically, just to be absolutely certain and be sure that you haven't missed anything. But the intuition gives you the first picture of what may be important and what will happen. Then you want to work it out in more quantitative detail, just to be sure your intuition is really right. You might have missed something. But by reasoning with intuition, you can think much faster. You immediately see what is likely to be important and what isn't. You throw away certain avenues of possibilities. Well, I'm pretty sure that can't be, that wouldn't happen. Something else would happen, you say to yourself. You suddenly see something very surprising and interesting that might happen. Then you follow it up with more quantitative reasoning that would convince your peers. Are we on? Then a more recent oral history is part of the AIDS oral history series that David mentioned. It comes, in fact, from 33 oral histories in this series, the medical response to the AIDS epidemic in San Francisco, 1981 to 1983. Now, those are the very first years when a mysterious disease was being recognized in San Francisco, New York, and Los Angeles. Those were the three early centers. I interviewed physicians, nurses, and social workers active in these earliest years when the mysterious syndrome striking gay men was first recognized, and when San Francisco led the way in medicine, nursing care, and social services for patients with conditions later united under the term AIDS. They, of course, didn't know what it was in the beginning. Connie Walsey, and we're about to hear an extract from her oral history, was an infectious disease specialist at San Francisco General Hospital and among the very first physicians anywhere to treat AIDS patients. In this extract, she provides a picture of the issues and emotions circulating in the earliest years of the epidemic before anyone knew the cause was a virus. And there's one clarification. She'll mention Ward 5B. Well, Ward 5B is the AIDS ward at San Francisco General, and of course it still exists. So let's hope it works. You can hear my voice instead of hers. We had 2 dry runs with this, and it's still not working. I've studied technology, but I have no capacity in fixing things when they go wrong. I ask her, or I make a statement, well, another dimension is fear. You know, we've been talking along. This is part of a longer interview. This is obviously a fatal disease for which it becomes clearer and clearer that the cause is a virus. What are you thinking about your own personal safety? Wofsy, we didn't. I think I can accurately say we. I think we were naive. I think we were so into it that we felt invulnerable. Most people I know had nightmares. I don't think I did. I heard what I wanted to hear from what the CDC, that's the Centers for Disease Control, said. And I believed it. They were like mantras. Hughes, that it wasn't terribly infectious? Wofsy, if it was infectious, why wouldn't there be more infected people? Stuff that's just bullshit scientifically. Hughes, that's what you wanted to hear. That's what I wanted to hear. Hughes, what sort of precautions were you taking? Wofsy, what did I do about needles? I was careful with needles. I think I was more careful than some of my colleagues who I felt were very cavalier. People almost bragged, I think, about the number of needle sticks they'd gotten. I mean, really, almost that cowboy-like approach. I remember the big deal for me was we went the other way. You don't wear gloves. You don't wear masks. Each person remembers his own role. I remember an infectious control meeting in about 1984, and I wish I had the documents, where the infection control guidelines might have read something like this, quote, don't wear gloves unless you are touching the patient's blood. Don't wear a mask unless you are doing bronchoscopy. I remember at an infectious control meeting making this statement, which was hardly endorsed, quote, it's time to reword this and say, wear a mask if you're doing bronchoscopy or da. Wear gloves when you're exposed to blood. In other words, we were so focused on the belief, don't make gay men feel like they're lepers, that you were almost intimidated if you wore protection. You were intimidated. And the guidelines said, don't wear protection unless. There was a turning point where we said, stop this. It's time to protect. Wear gloves. Then I ask, was the big deal made by the patients or by your colleagues? Wofsy, by certain colleagues there in infection control. The patients thought it made real good sense to wear gloves for examination of most areas. Frankly, I could have worn gloves for the whole exam and the patients wouldn't have given a damn, because the way in which I touch isn't a separatist touch. That's what people feel. It has very little to do with latex. But it came from certain members of the Infection Control Committee. Quote, we do not discriminate against our patients, end quote. The nurses on what was then Ward 5B, the inpatient aid service, were incredibly lax, and you wouldn't dare have said, quote, take more safety for yourself. That would have been interpreted as, quote, be afraid of these patients. Hughes, so this was a cause in a way. Wofsy, it was a cause. We were rebels with a cause. Some of us were more rebellious than others. Can we do another one? All right. And then the last audio clip is from a series with Genentech. Can you do it? Oh, that's right. And it consists so far of 42 oral histories and counting on biotechnology and bioscience. The Herbert Heinecker, who is, I will read his extract because the tape is, he has a very heavy Dutch accent, and even if we could get our technology working, you probably wouldn't be able to understand it. Anyway, he was one of the earliest scientists adept in recombinant DNA, a technique for combining and replicating genetic material. In this extract, he speaks about a period in the mid-1970s when he was a postdoctoral fellow in the UCS laboratory of Herbert Boyer, one of the two inventors of recombinant DNA. In 1978, Heinecker joined Genentech, at the time only 2 years old, the company that is, and now a Bay Area powerhouse in biotechnology. In this extract, we are talking about the guidelines for recombinant DNA research, which the National Institutes of Health issued in 1976 as a partial response to considerable public concern over the safety and implications of the then new technique of recombinant DNA. Many of you will recall the recombinant DNA controversy of the 70s. The context for the extract, which is the question of whether the NIH guidelines in genetic is the question of whether the NIH guidelines in the United States and related restrictions in the Netherlands inhibited progress in genetic engineering research. You may see some parallels with the current debate over stem cell research. Two clarifications. We refer to a P3 lab. That means physical containment level 3, a safety requirement of the guidelines for laboratories performing recombinant DNA research with certain types of viruses and microorganisms. And Nature and Science, as probably most of you know, are two major journals in the sciences. Hughes. We have danced around the political and social context, which was escalating in the years that you were at UCSF, around two themes: the possible biohazards of this new technology and, at a slightly later date, the appropriateness of academic research being commercialized. How aware were you, if at all, before you arrived in San Francisco, that there were some questions developing around this new technology? Was it a discussion point in the Netherlands? Heinecker. I can't recall now much of an issue it was at that time how much of an issue it was at that time in Holland. I think there was no recombinant DNA work going on in Holland at that time. It was really only done by a very small group of people elsewhere who had the tools. Coming back to Holland after 2 years in the United States, I found myself in a situation that recombinant DNA research was almost banned. We were very worried about the potential dangers of recombinant DNA technology. A government guideline advised us not to be involved in recombinant DNA technology. It did not go as far as to forbid it, but they only wanted to do certain aspects of recombinant DNA technology. Hughes. It wasn't a question of building a P3 lab or whatever. One just didn't do the research. Heinecker. Right. Well, P3 labs were being built in Holland then, but I never felt that cloning from one bacterium to another was dangerous, although there was very little knowledge about exchange of genetic information between different bacterial species. Now we know that bacteria exchange information all the time. But in those days it was not known and people were cautious. That was the main reason for me to go back to the United States in short order. Hughes. If you were reading Nature and Science magazines, it was hard to escape the fact that there was a political controversy surrounding recombinant DNA. Heinecker: oh, absolutely. There were those guidelines, sure. Hughes: did the controversy engage you? Heinecker: engage is a strong word. I found it a real drag because it was putting a string on the research that could happen. For instance, when we did the work on somatostatin, we had to do the experiments in a P3 room. Hughes: I was thinking of the situation earlier than that, around the time of the Asilomar conference. You could have said to yourself, this controversial area of science, I'm not going to touch it. Heinecker: well, perhaps I was too naive. I found it very interesting, important research. I really felt it was giving us some answers. I never felt that I wanted to give up on it. Did you think that there was any basis for concern? Heinecker: long pause. Well, yes, we could see, for instance, having the bacteria produce a human toxin and then the chance that these bacteria would colonize your intestines, that we felt, my goodness, that could be a little scary. So,
hopefully we can.
All right, this is supposed to be a video clip. We never know. And it is with Marcy Darnofsky, and this is a very recent project. This is a project that is in the process, in the very early stages of documenting the stem cell initiative. And this cut from a video was in an interview done by my colleague Ann Lage with Marcy Darnofsky, and Darnofsky is the Associate Director of the Center for Genetics and Society, an Oakland organization focusing on issues surrounding the new reproductive technologies. Darnofsky is not opposed to stem cell research, but you will hear that she has concerns about the social and ethical implications of the research. I am about to interview Paul Berg, the Stanford professor and Nobel laureate, whom I expect will present a somewhat different view from Darnofsky's. Please also note in this clip one of the strengths of oral history, how it relates personal background to the individual's later concerns and viewpoints. And then one point of clarification. Darnofsky will refer to Lee Silver, who is a professor of molecular biology and public affairs at Princeton, and the author, well, author of several books, but in this particular case, they're talking about Remaking Eden, in which he refers to the gen-rich people whose genetic makeup has been artificially enriched, and the naturals, meaning the rest of us who have to get along on the genetic endowment we were born with. So here she is. Let's hope it works.
Your objective core? Is it a philosophical core?
I think it's a social and political core. The social and the core for me really is about equality. It's about going back to Lee Silver's vision of a world of the gen-rich and the naturals. That's not a world I want to live in. And I don't believe most people would. And certainly not most naturals. I think that the trajectory of the technology in the kind of society that we live in today, where we do have these market forces, we do already have huge disparities of health and social condition. I think certainly it's not inevitable, but if we were to unleash these technologies, I think we would be in very grave danger that they would develop in ways that Lee Silver describes. And I think, one thing to now look back at, maybe something I could have said when we were in the earlier period, is that I really see this as a new kind of eugenics. And I'm Jewish, and grew up well after World War II, but certainly as Jews in the United States grappled with the Holocaust, and that really started happening when I was 10, 12, in my early teens.
And it was something you were aware of and
Yeah.
was part of your family?
I didn't have relatives who died in the Holocaust, but I did get enough exposure to Judaism that it was something that was discussed and not well taught and not in a way that really allowed me to chew on it. It was so horrible, I think, that it wasn't until later that I could really come to intellectual terms with it. But certainly I have a visceral reaction to anything that looks like, it's a eugenics. And while many people say, oh no, this is nothing like what happened then. It has nothing to do with government coercion. Just to start with, it would be, free choice. It would be consumer choice, and nobody would be making anyone do anything. Well,
The net effect.
it is very different, and I think that we have really struggled with how to talk about eugenics in a way that, we're not saying that anybody is, that's doing biotechnology research is a Nazi. We're certainly not saying that, but I think that it is historically the fact that eugenics is a kind of shadow and specter that geneticists have to confront, whether they want to or not. And I think it's very important for genetic scientists, especially those working in the human areas, to know that history and to understand how what looked at the time in the early 20th and mid-20th century like good science, in retrospect we call it pseudoscience. Some of it was people in the early 20th century climbing out on limbs where they really oughtn't to have been, but I think that's also true today. So the social consequences, the core is, the potential social consequence of going down a road toward this new eugenics that would exacerbate the inequalities and maybe lead to new kinds of inequalities and new kinds of discrimination.
Thank you very much. I'm sorry about the technical problems.
I'm Dan Kevlis from Yale, and I'm here to speak on the subject of genes, disease, and patents, Koshlan community, and biomedicine. But before starting, I want to express my gratitude for being invited to join in this celebration of this marvelous institution. I'm sure Dan Koshlan was teasing when he said scientists only care about the future, they don't care about the past. They do care about the past. He just talked about the past. And they invoke the past often in justification of principles, of policies, of practices, and so on. So do we all. And what's important in invoking the past is to try to get it right. And without the kind of materials that the wonderful repositories like the Bancroft Library collect and preserve and make available to scholars, we have no chance of getting it right. And so my applause and admiration for the enterprise that is ongoing here in this great institution. To turn to my subject, in 1988, in a report on the emerging human genome, which was just starting up, the Genome Project, which is just starting up, the National Research Council called for keeping open the data that the project would generate. It declared that access to all sequences and material generated by these publicly funded projects should and even must be made freely available. The admonitions to openness, of course, express the scientific community's long-standing communitarian norms, part ethical and part practical, that the knowledge of nature is to be shared. But the project, that is the Human Genome Project, had hardly gotten underway before it felt a counter-communitarian jolt towards property rights and cash. This came in 1991 from J. Craig Venter, biologist then at the National Institutes of Health in Bethesda, Maryland. He proposed the wholesale patenting of human genes, of human gene fragments called express sequence tags, or ESTs. Although just 140 or 50 to 400 base pairs long, each was unique and served to identify the gene of which it was a part. Venter claimed that ESTs would have utility as diagnostic probes for genes, but he also seemed bent on using the fragments to gain control of the intellectual property in the entire gene that the EST identified, even though the EST would reveal nothing about the gene's function. Within a year, the number of ESTs covered by the Venter NIH application for patents had multiplied to almost 7,000. A lawyer for the leading biotechnology firm Genentech noted, if these things were patentable, there's going to be an enormous cDNA arms race. Much to the relief of most academic scientists and a sizable fraction of the biotechnology industry, the United States Patent and Trademark Office rejected the Venter NIH patent application on ESTs. But the episode reveals that from the beginning, human genomics has been torn between a commitment to communitarianism, that is the openness and sharing of the data that would come from the project, and an impulse to cash and property. Communitarians disparage the cash impulse as a major departure from a long-standing commitment to cooperativeness in seeking to understand the workings of nature. There is a good deal of truth in the assumption of communitarianism in science, but the assumption is also suffused with a good deal of mythology, a romanticization of scientific practices in the past. And here, in my particular context, is an expression or an example of the need to get it right. So first, then, a few words about past scientific norms and practices in the interest of throwing into perspective the issue of cash and community in contemporary genomics. Cooperativeness is a cultural value of ancient lineage in science. The search for truth is said to transcend the individual scientist, the local scientific group, even the particular national scientific community. Through the long 19th century, a number of cooperative scientific endeavors emerged in fields such as astronomy, geology, and geodesy. Scientists in these fields were concerned with global tasks, for example, mapping the heavens and the earth, tasks that could be efficiently pursued by melding the results of local effort into a cooperative global network. But competitiveness has also characterized fields that lent themselves to cooperative effort, and it has been perhaps even more manifest in fields that have not. For example, the branches of physics and chemistry that are grounded in the small individualistic endeavor. More than 50 years ago, the pioneering sociologist of science, Robert K. Merton, pointed out that at least since the 17th century, science has been marked by rivalries, some of them ferocious. I need remind you only, for example, that Galileo attacked competitors for stealing credit for his invention of the military compass and the telescope, and that Newton, who was periodically obsessed with getting proper credit, battled with Robert Hooke over priority in optics and celestial mechanics, and waged a sustained war with Leibniz over the invention of the calculus. Merton wrote that priority disputes have not been the exception in science. On the contrary, they have, in his words, long been frequent, harsh, and ugly. One has to think back only in more recent times to James D. Watson's memoir, The Double Helix, to be reminded that even without commercial incentives, the practices of science can be marked by aggressiveness, secretiveness, competitiveness, and even ruthlessness. Since the late 19th century, the long-standing propensity to personal competitiveness was compounded by the increasing utilitarian payoffs of laboratory science. Physics and chemistry fueled what is known as the Second Industrial Revolution that has continued through our own day. In branches of these fields, commercial competition penetrated academic science far more widely than it had hitherto. World War I, World War II, and the Cold War introduced additional powerful incentives to national scientific rivalry. The close interweaving of science and national security put many laboratories under the wraps of secretiveness, and it brought many of those that remained open into sharp competition with the West principal antagonist. Even so, cooperation continued in some fields, notably high-energy particle physics, the most prestigious and expensive area of that science in that day. During the Cold War and since, freedom characterized the exchange of information among high-energy physicists. They have achieved an astonishing level of integration now via the internet in respect of creating, evaluating, and banking data about the properties of elementary particles. Whence this exemplary cooperation and consensus The answer, according to a member of a British group, to quote him, particle physics data have no economic or strategic worth. In the life sciences, circumstances contributed to a strong anti-commercial orientation. Most university research, especially in the basic life sciences, long yielded little that was patentable, and of that, less that commanded significant, if any, market value. For example, the workhorse of classical genetics was, of course, Drosophila. Although fruit fly geneticists developed these creatures into standardized strains at the cost of much time and painstaking effort, no one attempted to profit from them. Similarly with bacteriophage in the middle third of the last century, which was also standardized and made widely available among phage group geneticists. Cooperation worked because there was little reason not to cooperate and many reasons to cooperate, including the prospect of professional rewards and the joint advancement of knowledge. Academic culture's resistance to commercialization was particularly strong in the life sciences related to health and medicine. The University of Toronto scientists who were responsible for the isolation of insulin in the 20s excluded themselves deliberately from shares and revenue from the insulin patent, assigning their rights to the University of Toronto for $1. In the 1930s, Harvard University promulgated the explicit policy that innovations in medical research arising from its laboratories must not be patented, or if they were, the patent should be given freely to the public. Harvard's policy now, I think you will agree, seems, to put it mildly, quaint. Molecular biology has been demonstrating for some 30 years that it is highly practical, capable of generating both products and profits. Academic institutions and entrepreneurial faculty, with strong support from the federal government and their own administrators, have used the technologies of recombinant DNA, gene sequencers, and research tools to establish an astonishing fund of new biomedical knowledge and products. They have also joined with entrepreneurs to establish the modern biotech industry about which we have heard something this afternoon, and the intellectual property protection on which it is built, including the commercialization of human genes. Commercialization sharply challenged communitarian access to genomic data. If Craig Venter had failed at the wholesale patenting of human genes through ESTs, he did seek successfully, as I'm sure everyone here knows, to capitalize on human genomics ultimately through the creation of Celera, his company, and fast shotgun sequencing. Celera's original business plan called for its data to be held as proprietary by the company and released at first only to paying subscribers, while patents would be sought on genes of interest. After the human genome sequence was completed in 2001, Celera did allow academic scientists to download data, but only on a restricted basis. That is, an agreement not to give the data to anyone else. Other firms in the United States and Europe have managed to achieve exclusive control over genomic databases. Perhaps the best known is the arrangement of Decode with the Icelandic government. The company enjoys exclusive access to the National Medical Database, an agreement it entered into with government in 1998 for 12 years. A number of critics, both in and out of science, have objected strongly to such proprietary arrangements, especially where it involves human genomic sequence data. Many have advanced an ethical argument, namely that the human genome is humanity's birthright, that it belongs to the entire human community and ought not to be privatized in any way. That ethical argument has been largely ineffective against the commercial drive, but consequentialist claims arising from the mutual self-interest of most genomic researchers have done a good deal to keep genomic databases largely public. Valuable models demonstrated how this could be done. Among them was the activity called acronymically CEPH, or CEPH, Centre d'études des polymorphismes humains, established in the 1980s in France with genetic material from French and American families that was made freely available to scientists constructing the human genetic map. There was also something called the Worm Breeders Gazette, a record of the worldwide effort to map and sequence and characterize the C. elegans genes, the roundworm genes, including their multiple mutations. The Worm Breeders Gazette shared data, methods, instruments, and stocks, including mutants in the tradition of Drosophila genetics and phage genetics. The worm model influenced representatives of the multinational human genome enterprise when they met in Bermuda in 1996 to strategize the project scientifically and also draw up rules for the treatment of data. Clearly a response to the growing commercialization of the genome, the rules stated, to quote from them, that all human genomic data sequence information generated by centers funded for large-scale human sequencing should be freely available in the public domain in order to encourage research and development and to maximize its benefits to society. The Bermuda participants urged that all primary genomic sequence data should be in the public domain. The publicly funded human genome effort in this country, which since the early 1990s had operated on an international scale, has of course undercut privatization by retaining its commitment to openness in databases. Since the beginning of the sequence phase of the Human Genome Project, all data generated by participants has been deposited in publicly available databases every 24 hours. By 2003, the human genome sequence, essentially complete, was posted on the internet with no barriers to use, no subscription fees, no obstacles, and you can find it on the internet now. A growing number of journals will not publish genomic articles without proof that the authors have submitted their data electronically to a central genomic database. Such as the National Center for Biological Information, which places no restrictions on the use or distribution of its genomic data. In all, then, the communitarian commitment in modern life science has remained strong and has kept genomic databases widely available. But it's important to remember or to be aware of the fact that despite the ubiquitousness of open genomic data, communitarianism and cash competitiveness have remained in conflict in human genomics. The key reason is the patenting of human genes. The Bermuda rules, like Ceph, recommend against patenting human gene sequences, but there is nothing inconsistent with disclosing data after a patent has been filed on the one side, there's nothing inconsistent with disclosing data after a patent has been filed, period. Patents, once issued, disclose data to the world. And even if academic and biotech scientists submit genomic data to the public databases, they are free to file patents on it first. Well, what is wrong with patenting human genes, you may ask? Well, one powerful answer lies in the consequences of patenting them on both the practice of research and on the delivery of medical services. In research, even in universities today, the expectation of patentability discourages open discussion of technical detail in the labs during the critical R&D phase before the filing of a patent. Then, too, patented genes are research tools, and such tools, according to a decision by a federal court in 2002, are controllable by the patent holder, who may, in fact, restrict and charge for their use because research is part of what the court called the legitimate business of the university, and is not exempt from threats of patent infringement suits, even if its research is basic, applied, open, and non-commercial. A human gene patent establishes what has been called a chain of dependency in biomedical research. The chain reaches to efforts to characterize the gene and its functions more fully, and to develop diagnostic tests based on it. It thus has a chilling effect on all research that may involve the gene. One firm patented, for example, patented a gene encoding the CCR5 lymphocyte receptor without any knowledge of the link of this receptor to HIV infection. When the latter, that is, the link to HIV infection, was established by another lab, the patent holder declared that it would enforce its patent against anyone making use of the discovery in the development of any pharmaceutical to combat HIV. In 1999, a survey of 74 clinical labs revealed that a quarter of them had abandoned a clinical test they had developed because of pending patents, and almost half had decided not to develop a clinical test because of the patent. In the medical service area, some gene patent holders have tended to insist that only they can conduct diagnostic tests using their gene. The practice threatens, among other consequences, to concentrate expertise in only a few centers, to fragment molecular medical services, to elevate the prices consumers pay for diagnostic tests, and to make doctors vulnerable for infringement suits. It also flies in the face of sound medical practice in that it can deny patients access to second and independent diagnostic opinions. Such threats are not merely hypothetical, as is evident from Myriad Genetics' management of BRCA1 and 2, the two genes known to dispose women to breast cancer. For various reasons, by the end of the 1990s, Myriad held monopoly control through patents and exclusive licenses over the DNA sequence of BRCA1 and BRCA2. Myriad demands, as a matter of business practice, that all commercial testing for the two genes be done only in its own lab. It will not license the test to anyone, with the result that a woman diagnosed positively by Myriad cannot obtain a second opinion from an independent laboratory, which is something that you might think a woman positively diagnosed would want, because the only so-called therapy for this diagnosis is mastectomy. Myriad has enforced its patent rights against various universities, a hitherto exceptional practice. In 1999, for example, it notified a researcher at the University of Pennsylvania Clinical Genetics Lab that she was infringing the Myriad patents because she had independently developed a test to screen for mutations in the BRCA genes, and to cover costs was charging her patients a fee, a nominal fee, to undergo the test. Myriad advised the university to halt the researcher's activities or risk suit. To meet criticism from academic researchers, Myriad did negotiate an agreement with NIH in 2000, whereby NIH-funded researchers would be charged only $1,200 per test instead of the usual $2,580, so long as the purpose was research. In exchange, Myriad would have access to resulting research data. Even so, Myriad's BRCA1 patent covers a sequence not only as a descriptor of the gene, but as a substance in and of itself in its mutant forms. It also covers the uses of the gene as a probe or a primer, and it covers its protein. Myriad's patent claims cover all and any diagnostic method that uses the gene, including those developed by others. The company's practices appear to discourage the development of scientific knowledge and medical technologies, and the equitable, not to mention the sound delivery of medical services. So how should we now think about the evolving conflict between cash and community in human genomics, as exemplified by Myriad and its practices in the BRCA genes? BRCA1 was identified by a scientist named Mark Skolnick of the University of Utah. He had founded Myriad Genetics, and the university granted the fledgling firm an exclusive license on the sequence. Skolnick, contesting the idea that DNA is information, insisted it is a chemical and must be treated as such for patent purposes. He has said, if you discover a new molecule, whether it's a pharmaceutical or a paint or a dye or a gene, it's a new molecule, you should be protected. Genetic patents really follow the model that's been set up in organic chemistry. The U.S. Patent Office has affirmed that view, saying that if genes are treated as are other chemicals, progress is promoted because the original inventor has the possibility to recoup research costs because others are motivated to invent around the original patent, and because a new chemical is made available as the basis for future research. The fact of the matter, however, is that human genes are not like any other chemical. No one can invent around a human gene, including the mutated form that causes a disease. Human genes are the only ones we've got, and as such they are what an economist might call natural monopolies. As a society, we exclude private property rights in some natural monopolies, say Yellowstone National Park or the Muir Woods. We allow private property rights. We do allow private property rights in other monopolies, say railroads or the radio spectrum, but we do not permit the property holders to use their rights of ownership absolutely or arbitrarily. Where natural monopolies are concerned, we regulate the property right with regard to trying to achieve some sense of the public interest. The time has arguably come, I want to say here in conclusion, to regulate the kind of property right, the intellectual property right, that is represented by a patent in human genes, and possibly in human proteins too. The regulation might take the form of compulsory or voluntary licensing or patent pools. It might even take the form of denying patentability to human gene sequences, which would then make them available to anybody for research into the gene, the development of diagnostic tests for it, which could be patented, and discovery of its functions and malfunctions, and the creation of pharmaceuticals based on the gene, which could also be patented. This is a position advocated by many scientists, patient groups, and medical practitioners, including the American College of Medical Genetics. The strategy would allow for the patenting of the test and the drugs while leaving the gene itself freely available for research. I suggest that here is a policy that we as a society and Berkeley as a leader in Bay Area biotechnology in its innovative phase ought necessarily and profitably to consider. Thank you.